Okay, in terms of lab work, let's make sure we're all on the same page here. So remember that I told you that those NRO tubes were a little sketchy because they were so old and that some of the compartments were dried up, and I don't particularly trust them. So what you need to do is someone in your group needs to send me the number of your NRO tube unknown, and then I will send you back pictures of your NRO tube results, and you should use that for scoring your NRO tube. Now you can look at your NRO tube you did too and see how it compares, but use the pictures that I send you for the scoring exercise and filling out that scoring sheet and identifying it. Okay? If you want to save your NRO tube, you should get them out of the incubator and at least put them in the refrigerator. Don't leave stuff in the incubator. If you inoculated anything on Wednesday in terms of your urine samples or unknowns or anything, they should come out of the incubator. Okay, they shouldn't be in the incubator for 6 days, so make sure you do that. So any questions? Okay, so what we're going to be doing for the next couple of weeks now is we're going to be dealing with the third line of immunity, line 3. Once we cover the third line of immunity, we're going to follow that up with 2 additional topics, time permitting. And one will be applications of immunity and using immune reactions in testing procedures. So we're going to talk about serology. And then, particularly time permitting, we're going to end up talking about what do we do with that kind of information if we do diagnose a disease using serological techniques. How do we treat it? And so we're going to talk about chemotherapy, and particularly we'll spend some time talking about antibiotics. So that's kind of where we're going to go for the next exam, are these particular topics. Remember that the next exam is during the final exam period, and it will cover just this stuff. It will not be comprehensive over the entire semester. Okay? Line 3 of immunity is the most complicated. It's also the most fascinating because it really does involve a whole bunch of cellular and chemical interactions. And we're going to have to try to dig into some of that to a certain amount of detail. What are some of the characteristics that are associated with this third line of immunity? Well, I just said, complexity is one. And one of the things that makes these interactions so complex is that there's a tremendous amount of both positive and negative feedback interactions that are occurring. Third line of defense is adaptable. Remember that the first 2 lines of defense, we constantly use the term innate, meaning this was something you were born with. Third line of defense is something that develops during your lifetime and has a different pathway of development depending upon the environment you live in and what kind of things you were exposed to. And so this is something that you acquire over your lifetime. Another thing that characterizes the third line of defense is a very high level of specificity. And what we're going to be talking about a lot when we talk about the third line of defense are antigen-antibody interactions. In terms of levels of specificity, it is very similar to the kind of specificity you get between an enzyme and a substrate. Okay. So a very high level of recognition between one molecule and another. Another thing that characterizes the third line of defense is tolerance. You don't want the third line of defense responding to every molecule out there, particularly those that are part of your cells. So we need some self-tolerance built into this particular system. If self-tolerance isn't fully achieved, then we can run into situations of autoimmunity, immunity against self, and that's problematic. And there are a number of autoimmune diseases, some of which are pretty horrible. And then finally, there's memory associated with the system. So once you have been exposed to the real thing, the real pathogen, if you survive that encounter, you likely will then be protected against future encounters. Medically, we've taken advantage of that particular idea by immunization and trying to expose you to harmless variants of pathogens, getting your immune system educated so that then if you do get exposed to the real thing, you will have a high level of protection.
Does the third line in some cases also compensate for lapse in the first and second line?
Elaborate. I'm not sure where you're going.
There's some mistake in the second line because I mean it's innate. So like if there's some receptor that didn't work out,
yep.
Will it make up for that?
It possibly, yeah. And that's probably something else I should add to the list that I told you before, is that the immune system entirely in its kind of totality is highly redundant. And that's kind of what you're talking about. And so there are backups to backups to backups. And so, yes, indeed, third line of defense can compensate for some defects in first and second line of defense as well. Absolutely. This first diagram that I have up here is a nice summary of the third line of defense and breaks the third line of defense down into several different events, each of which we'll kind of talk about in a global sense first. And we have been talking about this in a global sense already. But I think for immunology, and particularly the third line of defense, it always helps to come back and kind of look at the big picture, then get into some detail, then come back and look at the big picture again, a little bit more detail. You know, in immunology, you've got to kind of take a couple step forward, step back, to kind of get a feeling for how all of this complexity relates and how the whole system works functionally as a unit. So let's start up here at the top and look a little bit at big picture ideas about kind of the development of your immune system and how you acquire this particular protective system as you grow and age. So we've got stem cells in your bone marrow, which are going to be the precursor to all of our immune cells. There are two major populations of white blood cells that are produced. They're the granulocytic cells and the agranulocytic cells. When we talk about third line of defense, we really are considering both types of cells because the macrophages and dendritic cells are granulocytic and they're intimately involved in the process. But the two major big players in the third line of defense, which are the B and T lymphocytes, are the agranulocytes. So let's concentrate on B and T lymphocytes for a second. So this is, I'll review something that I've said many times before, but B lymphocytes are involved in what kind of immunity? What branch of immunity? Cell-mediated or humoral? Humoral immunity. And what does humoral immunity involve? Antigens are involved in stimulating a humoral immune response, absolutely. What's a product of humoral immunity? An important product. Antibody. Okay? So we need to start cranking out antibody. And what's that antibody do? How's that protect you? It marks pathogens and foreign entities like virus for destruction. So the antibodies themselves are usually not the killing agent, but instead alert other killing agents, here's something that needs to be dealt with. The junior high school kick me sign that's placed on those particular pathogens. That's the role that the antibodies play. T lymphocytes are involved in cell-mediated immunity. And remember that cell-mediated immunity involves a couple of things. One is destruction of host cells that have become damaged. and the other is producing cells which aid the B lymphocytes in producing their antibody. Those would be the T helper cells. So, one of the things that's going to be really important for us to get a good handle on is the specificity that B and T cell receptors play in interacting with particular antigens. So there are going to be molecules on the surface of both B and T cells that will be paired with a particular foreign antigen, much like an enzyme is paired with a substrate. They have a high level of complementarity. They fit together. They dock with one another. Okay? How many antigens are there out there in the world that your body could encounter? It's really impossible to tell. But estimates indicate, it's in the billions of numbers of different kinds of molecules that are out there in the world. What's really interesting is that if we create an entirely new novel molecule through organic synthesis and inject that into an animal, they will form an immune response against it. They will form an antibody against that particular new molecule. That's kind of crazy. How do they do that? They've got to have cells with a receptor that matches that molecule that didn't exist before. That's pretty crazy stuff. So one of the things we're going to have to talk about is how these cells, as they're developing, build or make these particular receptors that end up on the surface of those cells. And then those receptors are destined, if you like, to bind to a particular antigen if the body ever encounters that antigen. If it doesn't, nothing happens. So that process of how you build that receptor during B and T cell development, how we educate those cells to recognize antigens, is a really important idea. And it's also a very bizarre set of genetics that are involved in this particular process. So that's probably something we'll get to next Tuesday right before Thanksgiving. So where does the education of B lymphocytes take place? They're born in the bone marrow. Where are they educated? Bone marrow. Okay, that's where the process of building receptors takes place. What about for the T lymphocytes? Thymus. Okay. So once you have gone through this education process, you now have a receptor on your surface that kind of pairs you with a particular antigen for your lifetime. Now, what we're going to find out is that on the surface of B and T cells, B cells have B receptors, T cells have T receptors, but there are going to be other receptors on the surface of these cells as well. What do you suppose? Do you think a B cell has only one copy of a particular B cell receptor? No. Why not have a whole lot of copies so you can bind a whole lot of antigen molecules? Pretty conservative estimate indicates that there are about 50,000 identical copies of a particular B cell receptor on a particular cell. So the surface of the cell is just covered with these receptor molecules. Once you become educated, you're still considered to be naive. Meaning you know how to do something, but you're not doing it. You have the skills, but you're not working. So these naive cells are being released from the bone marrow and the thymus. And what they do is they tend to then migrate out into the secondary or regional lymphatic tissue and take up residence there. So some go to the bone marrow. Some go to the lymph nodes. Some go to the malt. Some go to the gall. Some go to the spleen. Okay? So the body becomes populated with these educated but naive B and T lymphocytes who then sit there and waiting. So if you've got B and T cells in the nodes, for example, those cells are pretty much dormant. They're not divided. They're just sitting there. But if they receive the proper signals, then they start dividing. And they form a clone of cells, all of which are genetically identical to one another. They're dividing by mitosis. Remember, mitosis gives you what you started out with? Okay, so it produces clones of cells that are the same. So what are the signals? Well, one of the signals is binding of a particular antigen to a particular cell. So the antigen that enters your lymph node selects which B or T lymphocyte gets activated. We call that process clonal selection. Now, usually a single signal is not enough to stimulate clonal expansion. Usually you need a second signal as well. And those second signals are usually chemical signals, lymphokines. And what do you suppose a cell is that produces lymphokines that signals B cells to start dividing and producing antibodies? Where do those lymphokines come from? Maybe like a T helper cell? Something to produce those signals? This is a really important idea that typically a single signal is not enough to activate a cell into action to start initiating an immune response. Because this is serious business, okay? And you don't want to respond inappropriately. Sometimes the analogy is given to the immune response and a nuclear response. Anybody know how it works in a submarine with carrying nuclear missiles? Can the captain of the submarine make the decision on their own, launch? No, they have to get verification from other sources. They have to get other input before that dramatic decision is made to launch. Well, that's kind of the way the immune system is, okay? Instead of launching that immune attack by a single B cell, seeing an antigen and going, looks threatening, let's start a response, it needs other signals of verification before it really starts to go down that particular pathway. So, sitting in the lymph nodes, we've got all these primed, educated B and T lymphocytes waiting to respond if necessary. Binding of the antigen is absolutely essential for that activation to take place. But again, typically chemical signals are involved as well. This antigen is usually presented to these B and T cells as opposed to just a piece of foreign molecule binding to them and stimulating them. So antigen presentation is usually a big part of activation of these B and T lymphocytes. So what's going on there? Okay, so in this compartment down here, we've got entrance of antigen into your body somehow. It could be ingested, it could be an injury, a tear in the skin, whatever. But you get these foreign bacteria, viruses, etc., accumulating in your nodes, your blood, your tissue fluid. And there they encounter the phagocytic cells. Particularly important are the macrophages and dendritic cells. And what do those cells do? They recognize these foreign antigens because they have general groups of molecules on their surface that say, this is bacterial in nature, or this is viral in nature. What are those general groups of molecules that the pathogens have that stimulate phagocytosis by these cells? Those are the PAMs. Those are the pathogen-associated molecular patterns. And what kind of receptors on the surface of the phagocytic cell recognizes those PAMs? Well, one of them we talked about were the toll-like receptors. Okay? So they recognize these general categories of molecules. Okay? So phagocytosis takes place. And during phagocytosis, the pathogen is chewed up. And if it is an antigen-presenting cell, pieces of that pathogen are displayed on the surface of that antigen-presenting cell. Now, one of the things we're going to talk about in a minute is that when we start talking about antigen-presenting cells, there are some receptors that are really important in addition to the toll-like receptors. There are some receptors that are really important that we'll get into in more detail in a minute, called MHC receptors. There's MHC class 1 and MHC class 2. And they're really involved in this showing the antigen to the naive B or T lymphocyte. So, in this particular scenario, what we have going on up here is we've got an antigen-presenting cell showing antigens to a T lymphocyte that is educated and naive, but not doing anything at the time. And the presentation of that antigen begins to set a series of events and sequence that causes differentiation of the T cells into helpers and killers and things like that. The helpers start secreting lymphokines, which affect B lymphocytes, which in the meantime have also responded to the antigen and become activated. So the B lymphocyte's getting a signal by binding the antigen, and it's also getting a signal from the T helper, which has been presented the antigen. So this one's getting, proper signals now to take off and start doing something. What does it do? It divides. And when it divides, what does it do? It starts differentiating into different types of cells. We indicated a couple weeks ago that there are different pathways of differentiation for B and T cells. The T cells differentiate into one of three types: the helpers, the killers, or the suppressors. There are also memory variations of those particular cells. So we can have T immunological memory, cell-mediated immunological memory. The B cells differentiate into memory. Their effector cell is the plasma cell. And what does the plasma cell do? It starts cranking out antibody. Notice that these effector cells that start cranking out antibody no longer have the receptors on their surface.
I'm confused because I thought when you said they divide like to be identical.
Each one of these cells that is stimulated produces a clone of cells. These are subtypes within that particular clone. So a memory cell has the same genes as an effector cell, but they're not all being expressed at the same time. So these are genetically identical to one another, but this one is expressing different genes than this one is. This one is not expressing the genes for antibody production, for example, at that time. This one is. Okay? So look at these effector cells here. They no longer have receptors on their surface. Well, why not? Because they don't need to bind to antigen anymore. They've already been stimulated. And what do? They produce antibody. And is it a coincidence that the symbols for these antibodies are similar to the symbols for these receptors? No, not at all. As a matter of fact, the antibodies that these cells are secreting are very similar in structure to the receptor on the surface of the cell that stimulated the cell to produce antibodies. Come back. So, what happens? These cells start cranking out antibody. And the antibody enters circulatory system, gets in the lymphatic system, and marks pathogens for destruction, so that things like phagocytic cells can more effectively phagocytize the pathogen. Or maybe that antibody activates the classical complement system and results in that cell being then lysed. There's a lot of ways in which producing antibodies can protect you, and that's something else we will talk about in a little bit more detail later on. And then we got these memory cells here now, sitting there waiting for the next exposure. And the beautiful thing about a memory cell is that the time it takes for a memory cell to switch over to be a plasma cell is really short. The amount of time it takes a naive cell to differentiate into a plasma cell is much longer. So that's why we want to immunize and get some memory in our immune system. So that's the big picture. That's kind of what we need to wrap our heads around in terms of an overall understanding of what's going on in this immune response. The rest of it is a matter of filling in some details here and there to kind of complete the picture. So there's some background that we need to dig into now. So we're going to take a look at the big eagle eye picture. That's what this is. And now we're going to drill down into some little more background things before I think you can have a better appreciation for how all of this fits together. What are some of the background concepts that we need to understand? Receptors. We have to be able to understand the difference between MHC receptors, CD receptors, B receptors, and T receptors. And also with these particular receptors, we will spend some time talking about the genetics of how we build these particular receptors. Because for these receptors, we have to be able to build billions of different receptors, because there are billions of different antigens out there that need to be interacted with. So that creates an interesting genetic problem that we want to address a little bit. So that's one concept. Another concept that we need to talk about are some cytokines. Now, we've already talked about cytokines in one context of immunity. We've talked about some of the inflammatory cytokines that are involved in immunity. But this is going to be a different set of molecules now that we're going to have to consider. So those chemical signals that most immune cells need to be stimulated into activity. Third concept we need to talk about are antigens. What makes something an antigen? What are the different types of antigens that are out there in the world? I've already made reference to it. A type of antigen on the second exam. We talked about super antigens on the second exam. That toxic shock syndrome antigen. So what are super antigens? What are heterophile antigens? What are allo antigens? Okay, so we need to dig into that a little bit. And then finally, antibodies. Because really that's what the third line of defense is all about, is making antibodies. So what are antibodies? What's their structure? How does a cell make them? Why do they help your immune system? What are the protective functions of antibodies? So I think really this is kind of the order in which we're going to do it. First thing we're going to do is dig into receptors and talk about cytokines, then antigens, and then antibodies. And once you've got this background information, we're going to come back to this, basically, again, and go back through and, put some more detail into this, particularly down here at the bottom. And this interaction between antigen and B lymphocyte, antigen presentation to T cells, and any interaction between T lymphocytes and B lymphocytes. So you can either produce antibody or you can kill some kind of a damaged host cell. So this is something we're going to kind of keep coming back to as you get more background and ability to understand some of the nuances of this complicated system. So this is just another set of diagrams that kind of shows you the big picture. I'm not going to go through that. You can look at that. It's just another way to kind of demonstrate this relationship that occurs between all these different cells, T helper cells, B cells, cytotoxic cells, antigen-presenting cells, and what the outcomes of those particular activation processes are. Okay, so cell receptors. Let's start out by looking at what some of those cell receptors are. In general, what are the roles of some of these cell receptors? Well, a really important role is to detect and then bind to non-self molecules. We typically refer to those non-self molecules as foreign antigens. Another important role is to detect self-antigens. And if you detect a self-antigen, hopefully, in this instance, you don't respond. You want a response if you detect a foreign antigen, but certainly not if it's a self-antigen. Not all receptors are involved in binding antigen. Some of them are receiving signals or messages from other cells. So there are receptors for particular lymphokines, just as there are receptors for antigens. What happens if a particular receptor gets activated by binding an antigen or by binding a chemical signaling molecule? Well, typically something binding to a receptor activates some kind of a chemical signaling pathway within the cell. It's called a signal transduction pathway. So binding something to the receptor on the surface of the cell then initiates a set of reactions that translates into something unique happening within the cell. So the signaling molecule doesn't itself have to get into the cell. It's just binding to receptor, and then that sets off a cascade of events which results in something interesting happening within the cell. And one of the things that typically happens as a result of this is that there is some alteration of gene expression occurring. So the signaling cascade pathway turns on certain genes, maybe turns off certain genes. But the cells start doing something differently than they were doing before. So if we've got a naive, educated B lymphocyte sitting in a lymph node, and it gets the proper signals by binding to antigen and then binding to the right lymphokines, that activates those signal transduction pathways, and changes occur in transcription within the nucleus of that cell. And it starts turning on what were dormant genes for antibody production. So these receptors are really incredibly important. Some of those changes in gene expression alter cellular development. So a change in gene expression that occurs in B lymphocytes down here involves turning on genes for antibody production. But another change in gene expression that can take place is stimulation of a new developmental pathway that causes memory cells to be produced as opposed to the plasma cells. So this chemical signaling stuff is really, really important. Really, really crucial. Okay, so let's take a look first at MHC receptors. MHC stands for multi-histocompatibility. These particular receptors were first discovered in the context of tissue transplantation. Because remember that there are some instances where you can safely transplant tissue from a donor to a recipient, and the recipient will accept the transplanted tissue, but many times the recipient will reject the transplanted tissue. Why is it rejecting it? Because it is determining that that transplanted tissue is foreign and is mounting an immune response against it. If it accepts it, then it's recognizing it as being close to self. The molecules that help to determine that compatibility are these MHC molecules, these multi-histocompatibility molecules. MHC1 are found on all cells in the body, with one exception. Red blood cells do not have MHC1s. What's another way in which red blood cells are uniquely different from other cells in the body? They're non-nucleated, right? So they lack MHC1. MHC2 receptors are only found on certain types of cells. They're found on macrophage and dendritic cells. They are found on T helper cells. And they are also found on B lymphocytes. As it turns out, all of these types of cells, under the right conditions, can serve as antigen-presenting cells. So these particular class of MHC2s are going to be involved in antigen presentation. Now, these MHC2 molecules are anchored into the membrane of the cell. So there are polypeptide chains that make this portion that sticks out from the surface of the cell, and then there's a portion of the receptor that anchors into the membrane. Both MHC1 and MHC2 receptors have antigen binding sites associated with them. MHC1 receptors are involved in displaying endogenous antigens. Remember, these are molecules, endogenous, that are located normally inside the cell. So if you have a polypeptide chain that you are normally making as a cell, does that polypeptide chain last forever? No, molecules turn over in a cell. They're eventually catabolized and broken down. And as those polypeptide chains that you are normally making are catabolized, pieces of those get inserted in the membrane and associated with an MHC1 receptor, and then that can move out to the surface of the cell, and so a cell is then displaying its own antigens on its surface. And what's that doing? That's marking that cell as saying, I belong to Ethan. And here are pieces of the proteins that I produce, and this is my signature that says, I am part of Ethan's body. And that is one of the things that your immune system will constantly be looking for, is that cell signature of cells that normally belong to your body, to leave them alone. So that's kind of remarkable if you think about it. These pieces of every different protein you make are displayed on your surface in the context of one of these MHC receptors. And so in this little groove right here is a little piece of protein A that you're just normally making all the time. But that defines you as being a cell of that particular organism. If that particular tissue is then transplanted into someone else, and the surface of that cell, of that transplanted tissue, looks really different from the cell surfaces of that recipient, there's going to be a disconnect there. And that immune response is going to go, wait a minute. That looks nothing like Ethan's cells. And I'm going to respond. What are the likelihoods in terms of candidates for transplantation that would more probably be recognized as being Ethan-like? Who can you most likely get tissue transplants from more safely? Relatives who share common genes. But is that a guarantee of tissue compatibility? Not always. Everybody's got minor differences from individual to individual, even among siblings. But your chances are greater probably of getting a tissue transplant match from a sibling than it is a complete stranger. Although there could be a very good match with a stranger.
And you don't know that until it...
Nope. I mean, if you're a candidate for transplantation, then your tissues have to be typed. And they type both the tissues of the donor and the recipient and see if there are significant differences. And there's a certain amount of tolerance that you can have in the matching or the mismatching. And if the mismatching is too great, it's going to be rejected. Now, what happens if your cells become infected by a virus or become cancerous? Then they're going to start producing different proteins, right? Will those proteins be displayed on the surface of those cells along with the MHC1? Yeah. And that's one of the ways the immune system then begins to recognize, uh-oh, this cell's gone bad. I need to respond to it now. I wasn't responding to it before. But because it's now showing foreign antigens displayed on its surface associated with these MHC1s, now I need to respond. So those are endogenous antigens. Okay? MHC2 are involved in display of exogenous antigens. So these are acquired not because the cell normally produces them, but because the cell is engulfing virus or bacteria. And then it dismantles those pathogens, puts little pieces of those foreign antigens in an MHC2 molecule and displays it on its surface. Okay, so there'd be an MHC2 here if this were a piece of viral or bacterial antigen that the cell had engulfed. So, does it make sense then that MHC2s are associated with the antigen-presenting cells? No, those are the phagocytic cells. So they gobble things up, show the antigen in the context of this MHC2 receptor. There's a phenomenon that's going to be important later on called MHC restriction. And it's particularly relevant to T cells. T killer cells are MHC1 restricted. So the only way you turn on a T killer cell is it has to see a foreign antigen on your cell surface displayed as part of one of these class 1 MHC receptors. It's the only way it's going to respond to that antigen. Is it can only see it or detect it if it's in the context of being part of this MHC1 receptor. T helper cells are MHC2 restricted. So they can only detect foreign antigen if it's in the context of this MHC2 receptor. Remember, only MHC2 receptors are found on the surface of those antigen-presenting cells. So T helper cells can't respond to many different kinds of cells. They can respond to a macrophage that has ingested foreign antigen and is displaying it, or a T helper, or a B lymphocyte. But a T killer cell can respond to any cell that has gone rogue that's displaying a foreign antigen because it's displaying it on the MHC1 receptor, and all cells have that except the red blood cells. Another synonymous term for MHC is HLA. HLA stands for human leukocyte associated. A lot of the work with MHC2 receptors was originally done with white blood cells. So these are sometimes called HLA receptors, or they're also called HLA antigens, because the receptors are proteins. And proteins can stimulate antibody production. And so a receptor can also be something that stimulates an antibody formation. Okay, so remember on Monday I'm going to be out at a meeting, and so Monday is your lab time, and so I'm going to leave that to your discretion about how you want to use that time. Dr. Morton said he would be around Monday morning during the lab period, the 9 to 10 o'clock period, if you need something or need some help. Tuesday we will then come back and have more lecture, and then you get some break time.